Hello everyone. Welcome to today's live broadcast, The Paradigm Shift in Clinical Microbiology. I'm Christina Jewell of Labroots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by Labroots and sponsored by Roche Diagnostics. As a global leader in healthcare, Roche Diagnostics offers a broad portfolio of tools that help healthcare providers in healthcare providers in the early detection, prevention, diagnosis, and treatment of diseases. In molecular diagnostics, Roche is driven by a vision of working with laboratories like yours to improve the medical value you offer in microbiology, virology, women's health, and genomics and oncology. Roche continues to meet unmet needs through investment in research, innovation, and scientific excellence with the goal of supporting your important role in improving patients' lives. For more information, go to www.usdiagnostics.roche.com. Here's how the presentation works. We want to hear from you. Questions, comments, and even answers can be submitted via the green Q&A button at the lower left of your screen. We'll try to get everyone, but if not, we will make sure to follow up with you by email. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button at the top right or the Q&A button in the lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page and follow the process of obtaining your credits. I would now like to introduce today's speaker, Dr. Stephen Cagas. Dr. Cagas is currently the Scientific Affairs Manager within the Medical and Scientific Affairs Organization. He is responsible for interfacing with clinicians and key opinion leaders to assist in commercialization of products within the Roche molecular diagnostics portfolio. Stephen has expertise in donor screening using both the Cobas S201 platform as well as the Cobas 6800 and 8800. He is also the leader, he is also the lead for the microbiology assays for molecular MSA. Stephen has worked at Roche for 5 years in several capacities. He worked directly with customers as part of global customer support by handling complaints as well as assisting customers on site. He has also worked as part of the Operations Investigation Unit to perform quality investigations related to all kits and components manufactured at RMS. Stephen has a bachelor's degree in biotechnology from Rutgers University and a PhD in microbiology and molecular genetics from the University of Medicine and Dentistry of New Jersey. I would like to now introduce Dr. Stephen Cagas. Dr. Cagas?
Hi, Christina. Thank you very much for that nice introduction, and I really want to thank Labroots for putting on this webinar. I think I'm the last speaker of the day, so if you made it this far, congratulations, and I'm hoping that you'll really enjoy my talk. So my talk today is titled MDROs and the Smarticles technology. I wanted to start the talk today with a quote, and I thought that this was a very good one to use. It was from the World Economic Forum in 2013, and it says that humanity has always been under constant threat from infectious disease. Globally, we are getting better at monitoring signs of health-related crisis and alerting each other. There are far fewer deaths from pandemics today than a century ago. And we live in a bacterial world where we will never be able to stay ahead of the mutation curve. A test of our resilience is how far behind the curve we allow ourselves to fall. I thought that was really apropos and it was good for this talk. So the way that the talk is set up for today, it's going to be broken down into 3 basic parts. The first is going to be a little bit of background on MDROs, or multi-drug resistant organisms. The second part is going to be a current technology overview of microbiology as it stands now. And then the final section is going to be on Smarticles, the new technology from Roche. So I thought that I'd start with a little bit of a story, and this story was presented on a couple of different media, but the story is of a 19-year-old from the Seattle area who actually got into a train accident that cost him his right leg. But that seemed to be only the beginning of his battle, because he then had to battle several antibiotic-resistant infections. And the quote that he came out with was, "My life consists of watchful waiting and praying that the infection, like some awful type of cancer, does not return." This type of comment has actually been, I've heard this from quite a few patients throughout the time that I've been working on MDROs, specifically MRSA. And we have a speaker advocate who often works with Roche named Janine Thomas, and she also had a very difficult experience with both MRSA and a different C. diff infection, which we're not going to touch on today, but this is something very similar to what she says. She often has to worry and changed her lifestyle significantly to deal with the fact that she has methicillin-resistant Staph aureus infections, and she wants to make sure that she doesn't get any more infections. Another quote, oh, this didn't come out properly, but there is the WHO antimicrobial resistance report. So the WHO published its own report, and their quote was that the problem is so serious that it threatens the achievements of modern medicine. That is the problem of antimicrobial resistance and the thought of highly resistant bacteria for multiple drugs. So let's talk a little bit about healthcare-associated infections and what we mean by this. There are approximately 6 million healthcare-associated infections, or HAIs, per year in the US, Europe, and Japan. And with that, about greater than 70% of those HAI-causing bacteria are drug-resistant to at least one drug. And in a general sense, these are some of the more common that you'll wind up, that could fall into this category of drug-resistant HAIs, including methicillin-resistant Staph aureus, or MRSA, vancomycin-resistant enterococci, or VRE, and carbapenem-resistant enterobacteriaceae, or CRE. So I wanted to throw a little bit of a timeline up and give a little bit more background on the idea of multi-drug resistant organisms and a little bit of recent history. What you can see on the top left, that is the cover sheet from that WHO report I referred to earlier. It says that governments around the world are devoting efforts to a problem so serious that it threatens the achievements of modern medicine. A post-antibiotic era, far from being an apocalyptic fantasy, is a very real possibility for the 21st century. In other words, all of the work that was done throughout the 20th century to develop antibiotics, we may be in a post-antibiotic era with the ability of organisms to become multi-drug resistant over time and to all of the antibiotics that we currently have in our arsenal. In 2013, the United States Center for Disease Control and Prevention, the CDC, sent out an urgent threat warning that their emerging infectious agents are resistant to all known antibiotics that they exist right now. And to combat this problem, the Obama administration in 2014 started the process of trying to get increased funding for antibiotic resistance, and they started a task force called Combat Antibiotic Resistant Bacteria, or CARB. They announced several prizes and developed a strategy to combat antibiotic-resistant bacteria. In fiscal year 2016, the administration asked for $1.2 billion, which is at least double what they received in 2015, to try and increase the awareness and testing and development of new antibiotics. However, they received only $1 billion of that $1.2 billion that they asked for. And for 2017, for fiscal year 2017, they again asked for a budget of $1.1 billion. So they're continually trying to increase the amount of money that's dedicated to this particular situation because of the seriousness of the threat. In 2015, the UK is also looking to, obviously this is a worldwide problem, so the UK was looking to develop some more information around it, and they put up a $2 billion innovation fund supported by David Cameron, by the Prime Minister, to try and talk about ways that the UK could increase the testing. The picture that you see at the bottom left is an article from the International Business Times, and it's an article speaking specifically about carbapenem-resistant Enterobacteriaceae at UCLA. This is a big problem a couple of years back, where duodenoscopes were not able to be cleaned properly, and actually they were transferring these antibiotic-resistant bacteria from one patient to the next after the scopes were actually cleaned in between, but they weren't technically sterile, so they were able to pass, in a particular area of the scope that was very difficult to clean, they were able to pass these bacteria from one patient to the next as they were used. So this was a big issue, and this brought a lot of attention to the idea of antibiotic-resistant bacteria. So what is the threat of antibiotic resistance look like for the future? So in 2014, there was a report done by the WHO, the Antimicrobial Review on Antimicrobial Resistance. It's called the AMR report, and they did some estimates for the future, and they looked at what it would look like if we didn't do anything to take control of resistance. And they know that currently resistance is already responsible, drug-resistant infections are already responsible for 500,000 deaths globally each year. But what they said is that they expect that to rise to 10 million by the year 2050 and have a cost of over $100 trillion in lost output if we don't do things to try and control it now. We're going to talk a little bit more about this idea of the pipeline of new drugs, or the lack of pipeline currently. But one thing to note in this particular picture, and we'll come back to this idea in a couple of slides, is that you can see that the 10 million people that are estimated to be dying of antimicrobial resistant infections would exceed that of cancer, which would be at 8.2 million. So that just puts it into perspective what it would look like for in 35 years. So again, this idea of it's called the discovery void, that we haven't discovered a new class of antibiotics since 1987. And the World Economic Forum estimates that it costs Europe about $2 billion a year to fight lost productivity because of antibiotic-resistant bacteria. And Americans pay about $21 billion a year to try and combat this issue. So obviously it's not just a health issue, but it's also an economic issue, what it costs both you as a citizen, but also the healthcare system in a general sense to try and fight these organisms. There are a couple of interesting areas to talk about with respect to multi-drug resistant organisms and the global burden thereof. There was an article published that said that there were several studies done, and if you look at the incidence of the use of antibiotics overall, that the use is actually going up, which will contribute to more antibiotic-resistant bacteria. So in 2012, they did a study in India where antibiotics do not require a prescription. The pharmacy sales of the drugs increased 6 times between 2005 and 2010. And they found that a study in China revealed that 98% of patients in a children's hospital were given antibiotics for a common cold, which we know is viral, and therefore antibiotics wouldn't be effective for that use. But constantly prescribing them can lead to more antibiotic-resistant bacteria in a place like a hospital, which is especially prone to something like that. In 2014, there was a Princeton study that noted that between 2000 and 2010, consumption of antibiotic drugs increased 36%, with most of those coming from Brazil, Russia, India, China, and South Africa. But a related problem is also the fact that there are not as many infectious disease doctors as there used to be that are available to combat this kind of a problem. What you can see here is that the number of applicants per vacancy for the U.S. medical residencies and fellowships, that infectious disease doctors are actually only 0.8, so we're below replacement level for our infectious disease doctors. And what you can find is that the reason for this is that infectious disease doctors were the lowest paid among 25 medical professions examined in a compensation report in 2013. 2013. And they also they also had the second lowest application rate because of the fact that they have long hours and relatively low pay. And a second issue that is obviously related is the fact that microbiology journals form a relatively small field. So the number of journals that publish on microbiology is relatively small. And a similar study also found that there are 5 times more cancer publications than there are publications on microbiology. So essentially, you can see where the publications are going. That's what's driving the science, and therefore we're going to wind up with a bit of a lack of information, both for the MDROs in general and the doctors to be able to treat them. So in this AMR review, what they did is they, at the end of one of the sections of the review, they said, okay, what are 5 initial steps that we can do to increase, to start the process of reducing antimicrobial resistance? And what they found is that what they decided on is these 5 steps. The first is to increase early science funding to tackle antimicrobial resistance. The second is make existing drugs go further. So what they're trying to do is take the drugs that we currently have that we know are working and try and basically lengthen their lifespan so that the bacteria don't become resistant to them so quickly, and then we can actually use them for a little bit longer. And by doing that, hopefully we can bridge the gap between the current drugs and any future drugs, which ties in nicely with number 3, which is support the development and use of relevant diagnostic technologies. They said if we had the right diagnostics, more patients would receive the right antibiotic to treat their infection, but fewer antibiotics would be prescribed unnecessarily. So it's basically treat the patient with the right drug at the right time. Number 4 would be invest in the people who will solve the problem. Again, the infectious disease doctors and researchers to help discover new antibiotics and help stop the spread of bacteria that are already there. And number 5 would be modernize the way surveillance of drug resistance is done and used globally. It's important that we are able to actually monitor what we're doing and what the drug resistance bacteria field looks like out there. We want to know how many bacteria are resistant and what the percentage is in a particular hospital or a healthcare facility. So that's also important that we are able to track the problem and look at it as time goes forward. So the key role for MDRO diagnostics would be to track resistant bacteria and prevent their spread. So essentially what we want to do is find them quickly, track them through time, and prevent their spread from one patient to the next. With that, you want to also be able to guide therapy for patients. So what that would look like is we want to be able to quickly rule in an effective therapy, which would be able to improve the use of today's antibiotics. So instead of trying to use some shotgun approach or just prescribe the next antibiotic on the list, we would want to target the particular bacteria with the antibiotic that we know would work. So if we look at the current world of microbiology as it stands, what we can see is that there are a lot of different segments of the microbiology continuum. So in each of these areas, you can see on the top, there talks about the type of testing that can be done, and there's an example in the center circle, and then a little bit more about it at the bottom. So in the prevention world, there's screening, such as MRSA nasal screening. And a key part of the infection control process, screening tests we use to evaluate inpatients when they walk in the door or before a particular type of procedure to determine whether or not there's a carrier for something like MRSA. And with that, we can either decolonize them or we can isolate them more quickly, therefore reduce the spread. Then there's single diagnostic procedures, which are where you have a suspected pathogen and then you are able to detect just for that single pathogen. Then you can have a panel, which are bacteria that often have similar symptoms, and you can test a whole wide range of those. So an example would be a gastro panel, where you could test 10 different bacteria that could be gastrointestinal bacteria, and try and determine which one of those it is. Then there's bacterial identification, such as a gram positive or a gram negative. And that diagnostic procedure is for identification of an unknown pathogen. So it's usually at the very beginning of the process. But one final thing you have to do is after you identify the bacteria, generally you want to do what's called AST, or antimicrobial susceptibility testing, so that you can determine which treatment is going to be most effective for the patient. However, we're going to use this slide for the next couple of minutes to talk about some different things. However, what you can see here is, this is from a paper from Nature Review. What you can see is that the treatment processes and the workflow can be very complex for processing samples for bacterial pathogens. The first is that you often have a wide series of samples. So samples can be from many different areas, including blood, body fluid, urine, or even sputum or feces. And those samples that are normally sterile, you would want to culture those on a rich medium, and that can take a bit of time. So samples that are contaminated with colonizing flora are often can present a challenge when you're trying to pick out the normal flora from any infectious flora. But many types of culture media are also used for all of these different different types of samples that you can collect. So these are often referred to as selective media. So the media that you're looking to grow something on will either enhance the growth of the thing that you're looking for, or suppress the growth of things that you aren't looking for. That can also take a couple more days. So if you start on a rich media, you have to grow that for about 24 to 48 hours, and then you move on to a selective media afterwards. If you're not sure about what that bacteria was, then that could take another couple of days also. Then there's, during growth, you can check for morphological appearance and density, and specialized knowledge for deciding pathogenicity. So likely pathogens are then moved through a complex pathway to determine impact and antimicrobial susceptibility. So once you find the bacteria, once you isolate the bacteria that you are looking for through both growth on rich media and then followed by growth on selective media, you then have to start your identification process, and that can even start with something as easy as morphological appearance or density of the growth. So those are the beginnings, and then the next stage, what you would do is something like a gram stain or a MALDI-TOF. So these are two general approaches. And gram stain is a biochemical test. I'm sure that most of you are very familiar with it. And MALDI uses mass spec for species ID before setting up susceptibility testing. So if you're doing gram staining, it takes it's going to take some time to do the stain itself and do the species identification. Whereas with MALDI, you get the species identification very quickly. However, the final stage is then categorization of groups in species is required for the susceptibility testing panel. So susceptibility testing is often only performed by large reference labs, as it's a little bit more difficult, but the susceptibility testing can be done, but typing might be very difficult to do in-house. So it might have to go out if it's a selected bacteria that you want typed. So the upshot of this entire slide and what we've been talking about for the last little bit is that this process takes quite a bit of time. So essentially, if you're looking to do this process over and over for all the samples you have, it can actually be a bit of a time-consuming process and something you may want to speed up a little bit. So some of the classic microbiology solutions that are familiar are both chromogenic media, which we know is inexpensive and it has decreased time to result in culture, but it's used primarily for HAI screening, urinary tract infection, MRSA. Then there are the rapid tests. They come in a variety of formats. Then there's blood culture bottles in which you're directly detecting from blood. And the final are panels and reagents for ID and AST, and again, they can be manual or automated, and they can be ready to use for collections of media for ID. So these are a little bit more on the advanced side in terms of their ability to do multiple tests at once. But if we look at the clinical diagnostic approaches, this is one that we'll talk about a little bit more. This is what you're going to wind up doing for something like an MRSA is growing it on rich media, then doing some gram staining, and then you could also grow it in selective media, which would be enrichment broth, which has a high concentration of sodium chloride. And what you can see in the middle, that pink plate, is chromogenic media. So when you grow it on chromogenic media, the sample would grow pink, and other things would grow either white or wouldn't grow at all. The final system that you could use, so traditional culture, if you were to use the traditional culture route, it's going to take about 3 days to do a sample, to work up a sample. If you were to use chromogenic culture, this might be a little bit faster, and it might only take you about 2 days. One of the benefits of using a molecular test is that it can give you an answer as to whether or not a sample, for instance, for MRSA, is resistant in about 4 hours. So this is a significant reduction in the amount of time that it's going to take to get you that result to be able to help the patient. And if we look here, again, we're going to talk about laboratory identification. There are the traditional culture methods, the chromogenic culture, the chromogenic media, and molecular identification. All, again, for this example, would be for positive identification of MRSA and staph. And what we know about this, if we look at the difference between the performance of PCR versus culture, oh, this was supposed to come up a bit differently, but I'm sorry. There was a study performed in 2009 where there were 250 qPCR positive, quantitative PCR positive, and 250 negative samples were tested by genome and 2 chromogenic media with both direct and enrichment culture. And there were 186 culture positives for MRSA. And what you can see by both direct and enriched culture, this is for both of those chromogenic types of media. One is called ChromeAuger and one is called MRSA Select, is that the specificity is quite high with these, but the sensitivity hovers somewhere in the 80 to 90% range. And that could be problematic. And what several studies have shown is that past performance has shown that PCR to be about 98.2% sensitive. So an increase in sensitivity of approximately 10 to 20% over these chromogenic media, and about the same specificity. So you're not going to lose any specificity just because you increase the sensitivity. But currently, what are labs actually doing? What does it look like out there in the testing world? Well, if you look at the CAP survey from 2015 for MRSA, what we were able to do is break it down by who's doing the traditional methods and who's doing molecular testing. And what you can see is that even with all of the data that I just showed, about over 50% of the labs in this CAP survey were doing traditional methods, whereas only about 48% were doing molecular testing. And we're going to talk about why that might be in a moment. So again, what are some of the strengths of using the classic microbiology workflow? What could be the problem with the current solution? Well, the strengths of the current microbiology workflow in terms of plates and such is that the cost of the material is inexpensive. Plates are relatively cheap. They provide phenotypic information on resistance, independent of mechanism and after subculture. And they only detect live bacteria. You put dead bacteria on a plate, you're not going to see any growth, obviously. But some of the weaknesses are that it's a long time to result. We spoke about that before, how it takes time to grow up these types of cultures. It's labor intensive with specially trained personnel required. You're going to need somebody that knows what they're looking at when they look at a plate. It's expensive to automate. There are several systems out there for automation, but they can be costly. And there's a low sensitivity for most pathogens. So we spoke about that in previous slides, that the sensitivity is not the best with chromogenic media in the culture plate. So due to its weaknesses, culture-based diagnostic tests have lost some market share to both immunoassays and molecular diagnostics. So ultimately, what would we like to have? You'd like to have a target, a product that's inexpensive, that provides a phenotypic resistance information, and that's both highly sensitive and could, again, have that increased turnaround time, so or decreased turnaround time, the rapid turnaround time. You want the information in the same shift. You want the results with high clinical relevance, obviously, And you want those results to be both easy to use and have low resource dependencies. You don't want to have to babysit the type of system that you're going to have. You want to be able to basically walk away from the system when you start. So that's now where we get into the idea of Smarticles and the Smarticle technology. So I'm going to talk again. So this is just talking a little bit about what the challenge is for MDRO diagnostics. So again, if we talk about those culture-based tests, they're labor-intensive with a long time to resolve. But what we didn't talk about are some of the drawbacks of the NAT-based tests. And some of those are that there are many different resistance genes that are popping up, and sometimes it's not the same resistance gene. So those resistance genes are under they're constantly changing under the selective pressure of the use of antibiotics and the use of targeting of certain genes. And what you can see is that sometimes the gene changes. So that's where that phenotypic result is really going to help you out independent of mechanism. You'd really like to be able to find a result that's going to give you a little bit more information on just whether the organism is resistant or not necessarily what genes it has, because if the gene changes or a new mechanism comes up, then you have to try and detect a new gene in the NAT-based test. So this idea of Smarticles, where does it fit in? So there's obviously, we talked about the traditional and we talked about the molecular. And in the traditional, there's the culture. And in the molecular, there's the proteomics and mass spec. There's genomics, which we can detect DNA or RNA, which is the NAT-based tests and next generation sequencing. And then there's this new group that we're looking to display or to talk about, which is live cell molecular, or it's a bioresponse is basically what we're trying to talk about. And the Smarticle technology enables a lot of those things that we were looking at for the target portfolio, the target product portfolio, which would be detection of live bacteria. It's a phenotypic test, and it's direct from clinical samples. So there's no sample processing needed, independent of the type of sample that you're using. So what is a Smarticle? So the Smarticle technology, it's basically, there are two components to the Smarticle. The first is the outside of the Smarticle, and it's a specific bioparticle that's engineered. So basically what happens is you could consider it almost like a capsid that encapsulates something on the inside, but it's specific to a particular group of bacteria or even down to the species level. So what we see at the bottom is that the outside of the bioparticle can be changed, and what happens when you change it is you can change the specificity of that Smarticle. So you can get it from the family level for all of Enterobacteriaceae, for instance, or you can go down to the genus level, or even you can get it down specifically to the species level, for instance, something like Staph aureus. And so that's the outside of the Smarticle. And what goes inside the Smarticle? What goes inside is a synthetic DNA plasmid, and inside of this plasmid, it encodes a reporter gene. So what we can do is we can monitor the accumulation of that reporter gene, and when these two pieces are put together, which I'll show in a moment, when these two pieces are put together, basically what happens is that we package that plasmid inside of that biospecific particle, that pathogen-specific bioparticle. And that's what you get, that's what makes up the Smarticle. So the Smarticle is the packaged system in total, so including the outside, which is specific to a particular pathogen, and the synthetic DNA plasmid, which is inside. So how do Smarticles actually work? So what they do is, again, there's the bacterial genome, and the Smarticle will bind to the cell surface specifically to only the cells that you're looking for. And for instance, if it were Staph aureus, it would only bind to Staph aureus, not Staph epi, not coag-negative Staph, or any other types of Staph, only Staph aureus. And when the bioparticle binds to the cell surface, it injects that DNA, and it's able to transfer that DNA into the bacterial cell. And during that time, when the DNA is transferred into the bacterial cell, the DNA is transcribed, and the reporter gene it encodes for an enzyme, which the enzyme then accumulates throughout the matrix that the sample is in, and over time, during an incubation period, the samples are incubated and the reporter is allowed to accumulate. And finally, right at the end, there is the addition of a substrate. And when the substrate is added, the reporter acts upon the substrate and generates light, and we're able to detect that light that's generated by the addition of the substrate and the cleavage thereof. So the Smarticles cause the only living cells of target bacteria to produce light. So it's a true measure of the organism's response to antibiotics. So another level that you can do with Smarticle technology, which I'm showing here, is we're going to spend a second with this slide because this is actually the crux of the entire process. What you're going to do is you're going to add your clinical sample and your Smarticles directly to your clinical sample. There's no processing necessary, and the Smarticles will only bind to your target organism. So the Smarticles in this graphic are the small blue circles. The target organisms are the shaded, and the non-target organisms are the background color. So the Smarticles will only bind to your target organisms. They will not bind to the non-target organisms. And one thing you can do is you can add antibiotics to this media that they're incubated with. And if the bacteria that are present in that sample are susceptible to that, the bacteria will not be able to grow. Therefore, they will not be able to produce light. And if they're not able to produce light, when the substrate is added, you will not see a signal. However, when the antibiotic is added, and the antibiotic is if the bacteria are able to grow in the presence of that antibiotic, that means the bacteria are antibiotic resistant. And when you add the substrate, you will see light, and you will get a signal based upon that addition of substrate. So there are different levels of specificity involved in the Smarticle technology. So it's not just the abilities that I've spoken about earlier. There's 3 levels, and I actually think there might be a 4th level, but there's the cell surface, which we spoke about. So the cell surface specificity will only happen with the target organism. So again, if the receptor is not there for the target organism, so you can see here on the right in the blue, the receptor and the Smarticle in black don't meet up, so the lock and key are not combined, and therefore the Smarticles will not bind to that particular bacteria. That's level 1 at the cell surface. Then level 2 is actually inside of the cell, in which we can then target a particular promoter that would be specific to the organism of interest, which adds another level of specificity. So if we were able to target a particular organism, we're able to also target the reporter that we're going to use in that organism to turn on the reporter gene. And obviously the 3rd level would be the formulation, which you could add particular antibiotics to that formulation. The 4th level I spoke about briefly was this idea of live cell versus dead. So Smarticles, although they will only bind to the organism of choice, one question that often comes up is, well, what about dead organisms that may be present in your sample? Well, the Smarticle won't be able to, the reporter won't be able to produce light in the presence of dead organisms because there won't be the ability of the organism itself to generate more of that reporter gene. So it won't be able to, the reporter enzyme, so it won't be able to create light when you add the substrate. So there's not just the cell surface and the intracellular along with the antibiotics, there's also live versus dead. So only those particular organisms are alive of the particular grouping that you're looking for, whether it be species or genus, that have the particular reporter that you're looking for, and can either be susceptible or resistant to the antibiotic that you add to the media. That's the only ones that will produce light. So again, if we go back to this idea of MDRO surveillance, what the purpose is, it's again to track bacterial resistance and prevent spread. And how the Smarticles technology does that is by allowing the faster detection of these bacteria and being able to get that phenotypic result more quickly, which is exactly what you want. You want to be able to have that result, have it within that same shift that you're looking for, and be able to get the right treatment to the right patient at the right time. If you look at the way PCR currently works, when we detect one particular gene, the way the system works right now is basically you're only being able to rule out a particular therapy. So for instance, if you're detecting the MEK-A operon for MRSA, if you detect MEK-A, then you're saying, oh, you can't use methicillin-based drugs or beta-lactams. So what you're saying is don't use this. But with Smarticles technology, what you're able to do is actually say what you can use. So the bacteria that would not produce light in the presence of a particular antibiotic would be able to then give you the option of what type of antibiotic to use to treat a particular patient. So we could see that a sample would be carbapenem non-susceptible. So that would be a resistance, but you could use a different antibiotic that would work. So that's the idea. Instead of using things to rule therapies out, actually the most important thing with something like Smarticle technology is being able to rule therapies in for a patient. So if we go back to the idea of all these different systems right now that are out there for antibiotic, for bacterial identification and antibiotic susceptibility testing, what does it look like in terms of timing? So culture can get you your ID in a few days, as well as mass spec if you have your sample and you can culture it and get your identification. PCR can do that, plus a little bit of the resistance markers within 20 minutes to 6 hours, depending on the type of PCR. But a lot of these for antimicrobial susceptibility testing, turnaround times can take on the order of days to weeks to get a full susceptibility testing done on something. So the difference with Smarticle technology is that the identification and antimicrobial susceptibility testing can be done in, we're estimating, for approximately 4 hours. So not only do you do your identification of what's of your particular pathogen of interest in your sample, but you can also have an antibiotic susceptibility answer in that time as well. So to sum up about the Smarticles technology, what we find is that it combines the best of both worlds in terms of NAT and culture. You can do stuff, you can get you can do your testing direct from sample, you can get same shift results, and the answers are phenotypic. So the Smarticles technology has the simplicity of setting up a NAT test with the same speed, whereas the simplicity and speed obviously are not benefits of culture, but the phenotypic result is a big benefit of culture, and it's not something that we particularly have with a NAT result. But the Smarticle technology combines both of those into a nice little package that we're hoping can actually cause this paradigm shift in clinical microbiology. So with that, I'd like to conclude and say thank you again, and I'll turn it back over to Christina.
Thank you for that presentation, Dr. Cagas. We want to get right to your questions and input, so here's a reminder on how to let us know what you have to say. Questions, etc., can be submitted via the Q&A button at the lower left. We will try to get to all of you, but if not, we will be sure to follow up after the broadcast. Okay, Dr. Cagas, our first question is, so how do you address this concern regarding infectious diseases, physicians, and the growing awareness regarding HPV vaccination?
So the Smarticles technology wouldn't necessarily be applicable. For HPV, it's not particularly something that was in the pipeline of what we were looking to use the Smarticles technology for. Now, in terms of the actual menu that we were looking to develop, as the assay and the system are both currently in development, I can't necessarily disclose what the menu would look like. But suffice it to say that we are working on a pretty extensive menu for the Smarticles technology, but currently we don't have any information, and I don't think there is anything in development for HPV testing.
Stephen, does the increase in antibiotic prescriptions in Brazil have a link with time lapse between consult and antibiogram delivery?
So the time difference between consult and therapy initiation could be a reason for increased antimicrobial resistance. However, studies have shown that the time between isolation, so what you want to do if you have somebody who has a resistant organism is you want to get them, you want to get that organism identified, and you want to get it treated as quickly as possible. So, and you want to isolate that patient as quickly as possible. So, for particular tests that are, so for inpatients, generally what they'll do is they'll isolate the patient if they know that they're either an MRSA carrier, or they'll try and get the result within a certain amount of time to try and then triage that patient and start to do that in a different way. In terms of the reason for increased antibiotic resistance in Brazil, there may be a causal link between that amount of time, but I'd have to look that up a little bit further.
Next question. With respect to molecular genetics, are there plasmids or constructs that could utilize this Smarticle technology for mechanistic studies?
So this is actually a pretty interesting topic in terms of the ability to use Smarticles outside of the particular way that they have been designed up to this point. However, at this point, the Smarticle process and the Smarticle technology is currently only for use for the particular assays that we're looking to develop. And I don't think that Roche at this time is looking to license that technology or move it forward to be able to do studies with it. Perhaps when the FDA clearance comes for the technology, there will be different ways that researchers could use the technology for different types of studies and all different things that I'm sure people are already thinking about in terms of different sample types and in terms of different collection systems. So.
Next question. Specificity of smart bioparticle is negative 99% as checks 3 area. What's the sensitivity and which Roche instrument diagnosed MDRO?
So for the Smarticles technology, we are working on a system that would be we're working on a system that would be specific for the use with the Smarticles technology. What we would like to do is be able to present that information when we have clearance for both the assay and the system. So as of right now, I can't give you much information about the particular system that would be running the Smarticles technology. But I do look forward to giving another presentation where I can give you much more in the way of specifics on both the assay menu and the system itself that we're looking to develop for this.
Okay, in terms of costs, how the new diagnostics approaches can provide more benefits in comparison to culture besides the economy of time.
So, yes, so certainly there are other benefits to the new diagnostic approaches other than just the economy of time. Certainly the time factor is one that would be helpful as the ability to answer the question and be able to triage the patient. Is certainly the time sensitivity is also very important. However, we would also want to be able to increase the sensitivity and specificity of the systems over the traditional culture types. So one of the things about the culture systems right now is that their sensitivity is actually quite low. And with something like NAT or nucleic acid-based system, or even the Smarticles technology, although I didn't present any of that data, they're much more sensitive than traditional culture-based systems. And also with the type of samples, there's often you have to do the enrichment if you're going to be doing culture, and that can also take time, as well as some other things like the requirement of a technologist to be able to understand that culture plate and read it and interpret it. The Smarticles technology and the system that we're looking to put forward doesn't have any of those restrictions on it. That would also contribute to the benefits other than just time.
Dr. Cagas, we have time for one more question. Can you give us an idea about the menu Roche plans to develop using the Smarticles technology?
Yeah, so that's the $1-million question. The menu in general at the beginning will be somewhat limited, but Roche is actively pursuing ways to increase the menu and things that we think are most appropriate to use this technology. However, one thing that's very important is there are a bunch of healthcare providers and a bunch of very smart people that are probably listening to this. So if you have suggestions on what you would like Roche to pursue, or what you think would be appropriate avenues to use the Smarticles technology, we would love to hear from you and understand what you see in your particular healthcare facility every day and what you think will be the most important, whether it be a particular bacteria that you think would be very important to be able to identify, or a particular bacteria/drug combination that you think would be important. So stay tuned. That's the short answer, or the long answer.
Well, we would like to thank you again, Dr. Cagas, for your presentation. Would you like to leave us with any final comments?
Sure. I'd really like to thank the audience. I appreciate you guys listening to my talk and understanding the Smarticles technology. I would also like to thank Labroots very much for hosting this and for having me And again, we'd really like to hear anything you guys have to say about the Smarticles technology and where you think it could best be used. So thank you very much, and I appreciate all the feedback.
Thank you, Dr. Cagas. And thank you again to Roche Diagnostics for sponsoring today's presentation. If we weren't able to speak to you directly today, we'll be sure to follow up with you via email. Thank you for taking part. We want to thank today's presenters as well and for joining us here at Labroots.com, the leading scientific social networking site. For more information, visit us at www.labroots.com. Today's webcast will be available for on-demand viewing on our website through December 8th, 2016. We will let you know when it's been posted, and we hope you pass that information on to any colleagues who couldn't take part in today's broadcast. See you next time. Goodbye.